Yep, so hi, I'm Kenny Delgado. I'm CSO of Jetbio, also Professor of Manufacturing Engineering at Newcastle University. And what I'm going to do is talk through a potential application area for Jetbio, which is looking at models, in vitro models of leukemia, so that we can understand the interaction between drugs and leukemia cells and the development of the cancer. Okay, so I'm going to try and do three things. I'll sort of introduce our bioprinting technology, describe research on modeling of drug resistance in leukemia, and then explain why our printing technology is very well suited to developing these kind of state-of-the-art models for in vitro testing and drug development. Okay, so our bioprinting technology, it's patented, it's with Jetbio and licensed to us from Newcastle University, where we developed it. And it's a way of producing cell-filled gel cultures. So cells that are suspended within a gel culture. Gel culture is quite attractive as a way of doing cell culture for drug models, because it puts the cells in a sort of biomimetic matrix. Gels can be developed to have kind of properties that are quite close to those of tissue, and so the cells have a kind of a matrix surrounding them that's similar to what they would see in the body. And so that's so cell-filled gels is what we're normally looking at. We have a particular way of making it, and that's what we have the patent around. We generate two droplet streams from two microvalves, and one of them's a polymer droplet stream, and the other one is a cross-linker droplet stream. And we arrange for these droplets to meet, and where they do meet, the two materials react to produce a gel, like jelly, or whether shower gel or jelly that you eat or whatever, but that kind of consistency. And what it looks like in practice is this. So here you have two microvalves here, and you can see the gel being deposited onto the substrate down here. A bit in the middle where the droplet streams are and where they meet kind of happens a bit too quickly, and the droplets are a bit too small for you to see, but that's what's kind of going on in there, and you get the gel deposited. In order to show you the droplets meeting, we need a high-speed camera, and this is kind of what it looks like, and we've done some mathematical modeling of that as well to understand the mixing of the droplets. And you've seen us on the previous slide do a droplet array. We can do kind of 2D structures, 3D structures by putting layers on top of one another, and we can pattern those into shapes as you can with any kind of bioprinting technology. And so that's what we do. And then, so that's how these are printed gels. They're not cell-filled. If we want to print cell-filled gels, what we do is we suspend cells in either the polymer or the cross-linker solution, or in both of them, and then those cells get encapsulated within the gel as it's dropped down onto the substrate. Okay. And what a cell-filled gel looks like if you print it is this. And so the white you can see in here is cells, and here this is this gel that was printed here has 10 times the number of cells that this one has, and so it's more kind of opaque and see-through. With confocal microscope, you can see that we've got more cells here than we do here, and that they're closer together. And that's one of the kind of high cell density is one of the kind of key advantages of our bioprinting technique. This study, we were looking at bioprinted bone microtissues, so we were printing stem cells and then differentiating them to become bone cells and tracking how the tissue matured. The little nodules that you see in these two pictures after 14 days, those are calcium deposits that the bone cells put down as they're maturing to kind of build the tissue. And what we find is that we get a lot more calcium and a lot more bioactivity from our high cell density gel than from our low cell density gel, and that's not just linear. It's not just that because we've got 10 times the number of cells we get 10 times the output, Actually, under some measures, we get 50 times the output. So you actually get more output per cell as a result of the cell having lots of other cells around. And so that's quite valuable for us. Okay, so that's yeah. So those are just some examples of cell-filled gels that were printed and the value of high cell densities. If I kind of move on now to think about leukemia, we've been working with Dipali Pal at Northumbria University in particular. She leads this study, and Sean Hockney is the research student who's been doing most of the work. And this is work where we're looking at co-cultures for leukemia. And this is some work that's just about being published. This is a preprint on bioRxiv that's gone up earlier on this year, and so it's new results. And in terms of the kind of the overall aims, clearly improved and low toxicity treatments are needed, and we need ways of understanding what they should be. But we don't actually have the in vitro models available to us to accelerate cancer drug development in the way that we would like to at the moment. And so that's the overall aim of the project. It's funded by the NC3Rs and, as I said, is led by Dipali. And then what we've started off with was a model development pipeline where we want to end up with a co-culture, as you see over here, of stem cells, which are cells that are resident in bone marrow, and leukemia cells, which are clearly the cancer cells in this case. And so we started off looking at the gel itself, the gel with leukemia cells around and invading it, then looked at MSC cells, the stem cells on their own, then co-culture, so we could start to track the relationship between the leukemia cells and the stem cells. And then longer term, clearly we want to build the model up in a scalable way. And so the idea was that our bioprinting system would be able to produce lots and lots and lots of these gels once we were very clear about what the model was actually focusing on. And I'll say a little bit more about that in a sec. But where we are now, thanks to Sean's very, very good work, is we're up here looking at the co-cultures. Okay, so here I'm just going to zoom in, if you like, on the co-cultures and let's see what's going on with them. So this up here, this is a co-culture. Green is a stem cell that resides in the bone marrow, if you like. The red is the leukemia cells. It's acute lymphoblastic leukemia cells. And so we have them in a co-culture. We can look at them through different filters to see them separately, or we can see them together if we decide to do that as well. Okay, and so this is the cells within this co-culture, and this top line is showing you just the cells in a co-culture being cultured, nothing being done to them. This bottom line down here is what we're introduced to dexamethasone, which is a steroid which is used in the treatment of leukemia. And when we introduce the dexamethasone, we start to see these little fibers, these fibrils, happening between the cells. And these are the stain in the cell shows us the transfer along these fibers. They're tunneling nanotubes. And what they show us is that when we introduce this steroid, which acts against inflammation and may act against leukemia as well, it kind of stresses the cells and causes them to exchange material from the cell nucleus, which contains DNA, which allows, so there's transfer of DNA between the cells. And we can see it happens between the MSCs and it happens between the leukemia cells. And over here, when we look at both together, we can see some areas of yellow. So the red and green are kind of mixing, and we're seeing something sort of different going on. And what we think is happening here is that this is kind of evidence that the the leukemia cells are communicating not just with other leukemia cells, but they're communicating with MSCs as well. And so they're responding to the stress caused by the dexamethasone by starting to infect other cells. And that's really what it does. And so it's an example of how cancer cells show drug resistance, because they're always trying to be a step ahead in terms of how well they spread leukemia, so that the individual cells may die as a result of the treatment, but actually there are some residual cells that are there to pick things up again as we go along. And so that's what we think is going on here. It's really interesting to be able to see this process, and ability to see it's quite significant, but it gives us a really cool insight into how the leukemia develops its drug resistance in response to a threat. Clearly, if we look at dexamethasone, we can also look at other drugs, and so this is our drug ABT199, and you can see that actually we still see some of these tunneling nanotubes when it's introduced. Another drug is ADH1, and we see tunneling nanotubes from in it as well, to some degree in the MSCs, actually to less degree in the leukemia cells, which is a good thing. And then, because we have this nice scalable in vitro model, we can look at drug combinations. And so this is what happens if we put the dexamethasone together with the ABT199. You see nanotubes in the stem cells and in the cancer cells, but with dexamethasone and the ADH1, you see very little going on in terms of the transfer of materials between cells. And so these are lovely models which allow us to explore these things, and this is telling us that in this case actually the combination of the dexamethasone and the ADH1 is much better than any of the drugs on their own. And so this kind of drug combination would be a really excellent way of treating this particular strain of leukemia. Okay, so why then, as we scale this up, so those models are kind of handmade, we can print them as well, but why would reactive jet impingement be the right solution to scale this up? Well, first of all, you absolutely need high cell density. If we don't have high cell density, then the key cell-cell interactions are missed. We need these cells to be close to one another to see these interactions. Second is that you need a biomimetic gel. So these nanotubes are going through tissue and through gel, and so if we don't have the matrix that the nanotubes have to go through, again that's a very artificial situation, and so we have a much more biomimetic arrangement of things because we have this biomimetic gel, because it's a gel culture. Finally, yep, we've got where we are. We now need it to be an automated, scalable solution. If we want to look at multiple drugs and multiple doses, then that means we need lots and lots of samples. If eventually we want to be able to do this, clearly, ideally, you'd identify a universal solution, but it's possible that it needs to be personalized medicine, that you need to develop a drug combination for an individual. And that would mean, yeah, lots and lots and lots of samples. So you want to be able to do that effectively and cheaply and in an automated way. And the Regi bioprinting approach lets us do that. So just then, main takeaways. We're the only company with the reactive jet impingement bioprinting technology. That brings unique and valuable capabilities to the market. And what we're planning to do, not just in leukemia, but in lots of areas, is look at combining it with other biofabrication techniques to get technology solutions which underpin new drug development, as I said, precision medicine, and also we want to look at, and we are looking in other research projects at scalable approaches to tissue engineering using the technology as well. Okay, thank you.